So this is the last lab meeting that we have for this semester. Next week's lab meeting time is set aside for optional dissection, like I was talking about in lecture. If you didn't let me know by the end of last week that you wanted to do that, then next week's lab period is canceled as far as you're concerned. So you don't have to be here next week. What we'll be doing today is looking at the reproductive system. We'll be starting the reproductive system in lecture tomorrow, I'm pretty sure. And usually at this point in the semester, I'm running a bit behind. So I'm trying to squeeze a number of different things into the time I have left. And I have in the past used lab to concentrate on the male reproductive system and lecture for the female reproductive system. And since I've done that pretty habitually, the assignments that you have are based on that. So the lab quiz and the practical are mostly about male reproductive system, and the assignments you have for the reproductive system chapter concentrate on the female system. So we'll keep to that. Now, what I generally do in starting off with a reproductive lab is to explain sort of the purpose or function behind the reproductive system, and talk about a little bit there. Let me think here. So what I was thinking about is we actually have a little bit more time in lecture than I would think, or I would normally plan on at this point in the semester. But I'll go ahead and cover this in lab just to sort of be consistent with what I'm usually doing. Let me just point out in the book, this is the final unit of the book, which has just 2 chapters, the reproductive system and then development and inheritance. And some of what I want to talk about sort of to set up the reproductive system kind of comes from the development and inheritance material. I actually want to go to talking about it through the presentation that I have for this. There are a few pictures that come from other books, and I just haven't updated them to the current book's pictures, but there's equivalents in the book that will sort of complement what I'm talking about here. So giving us a second to load. And let me put off some of the lights. Now, the purpose of the reproductive system really is to reproduce the species, to make more humans. And so what I want to talk about is some things that kind of define the species biologically, and with that a little bit of just how we define species biologically across the board. The picture I have here is showing the egg cell which is the female reproductive cell, covered in sperm, which are the male reproductive cell. Now, this picture is actually a little bit misleading. This isn't exactly what it would look like in the real in vivo situation. This is probably made through processes similar to artificial insemination. So this egg's been exposed to sperm in a test tube or petri dish or whatever you want to say, and then it's been prepared for microscopy for the scanning electron micrograph so we can see this. What would really be going on in the actual situation, which I'll be talking about a little bit later, is a lot fewer sperm would actually be at the point of contact with the egg cell. Probably hundreds of cells that we see here is a little bit misleading. It's not usually that many cells that actually make it to the egg cell. Now, once the egg and sperm cells come together, then the fertilized cell starts dividing. So these pictures sort of show us the sequence through division. The fertilized egg cell cleaves into two cells. So the two big cells there are the divided fertilized egg cell. There are two little cells at the top called polar bodies, which we'll talk about in lecture a little bit more detail. But so that one cell divides into two, and then those two cells divide into four, and then four cells divide into 8, 8 into 16, 16 into 32, 32 into 64, 64 into 128, 128 into 256, 256 into 512, 512 into 10, 24, 1024 into 2048, et cetera, et cetera. That's just a progression of the powers of exponential powers of 2. So just when one cell divides and then each of those cells divide on down the line, after I kind of lost count, I think there was 8 or 9 divisions, we get from one cell to thousands of cells. That progression takes a little bit longer than what this picture is suggesting. This is 5 days worth of development of the very early zygote, what we have before the embryos starts developing. At the day 5 point, the group of cells that have been dividing exponentially, like I just sort of suggested, start to organize into different regions, which include an area, a mass of cells inside, which is the embryo blast. And those are the cells that will become the embryo. And then the embryo will eventually develop to the stage that we call it a fetus. And then the fetus completes gestation, and the newborn is what we obviously call something that was just born. Now, all of the cells that we see here and all of the cells that end up making that newborn baby are really just descendants of that original fertilized egg cell. And the cell division that's going on here is what's called mitosis. Now, mitosis technically is talking about nuclear division, and then it's followed by the cell dividing so that the two new nuclei each have their own cell. But people tend to just say mitosis is a type of cell division, which is a fine oversimplification for the sake of what we're talking about here. Within mitosis, what happens is the DNA within the nucleus duplicates and then separates into two equal groups so that the two resulting cells have the exact same DNA as the original cell. This is what we would consider cloning of cells. Now, when you hear cloning, people often get sort of a Hollywood idea in their minds, which would be a Arnold Schwarzenegger movie or something like that. But the word cloning really describes cells that have identical DNA. And so when scientists first cloned a sheep, Dolly, which I don't remember how long ago that was now, maybe decades. Really what it was is they took the DNA out of an adult sheep and put it into an egg cell. And then as that egg cell divided through all these steps, the DNA that it was copying and replicating was the exact same DNA from the adult sheep. So the resulting new sheep was just genetically identical to the first one. So that's what cloning really means. And in mitosis, the DNA is copied pretty much exactly. So every cell has the exact same DNA in it. Now, looking at that DNA is part of how we sort of define what the human species is. How many pieces of DNA do we find in a cell in your body, with a few minor exceptions? How many pieces of DNA would we expect? Or what do we call a piece of DNA? One complete DNA molecule in the nucleus.
Is it a chromosome?
Right, chromosomes. And how many chromosomes do we find in the nuclei of human cells?
26.
It's 46. You get a chromosome from either parent, so you end up with 23 pairs of chromosomes. So that 26, I'm sure, was just a combination of those two numbers. Then let me see here. Oh, sorry, this is just, that's right. So in those cells as they divide, the DNA is exactly the same, but at some point the cells start to differentiate. So all of the cells represented here, which really are kind of like three different types of cells, are genetically identical, but especially in the way that this is drawn, the cells are physically different. The different types of tissue that we see here, the endoderm, the mesoderm, and the ectoderm, those are just cells all working with the exact same DNA and expressing different genes within that DNA that leads to different properties of the cells. And we see that continuing on as we get all the way down to the adult stage. All of the different cells in our body, with a few exceptions, have the exact same DNA. And what makes them different is just which genes that they're expressing. So a skin cell, an epidermal cell, expresses the keratin gene to make that particular protein that's an important part of how skin works. Or muscle cells make myosin, which is a specific protein found in muscles. Or nerve cells make the voltage-gated sodium channel protein, something like that. But the other types of cells don't use those genes. So part of what really defines the human is the number of chromosomes that it has and the genes that end up being expressed. Oh, never mind that stuff. This is where I was going from. This is a picture that's called a karyotype. And it's just a picture of all the different chromosomes. Like I said, you get one of each chromosome from either parent. So there are two chromosomes number 1, two chromosomes number 2, et cetera, one coming from mom, one coming from dad, all the way through 22 pairs, and then the 23rd pair is the sex chromosomes, which in this particular karyotype is an X chromosome and a Y chromosome. which tells us that genetically speaking, this comes from a male of the species. Female would have 2 X chromosomes. The chromosomes are numbered based on their length. So chromosome number 1 is the longest. Actually, 21 is the shortest, but before they were really precise about measuring the length of the DNA, they thought that 22 was the shortest, so that's what got numbered 22. And they didn't change that because the number of the chromosome is an important part of knowing where certain genes are found. Now, really, the Y chromosome is the shortest, but it's not part of the pair. So, of the paired chromosomes, 21 and 22 are the shortest. 21 is the longest. The X chromosome looks like it's probably about the size of chromosome, maybe somewhere between 8 and 12 or something like that. And then the Y, like I said, is definitely the shortest. But the that 23rd pair, the sex chromosomes, are sort of set aside as special. Now, the point of the reproductive system is to deliver this DNA so that the offspring is going to get the exact same number of chromosomes as the original, as the parents. And so each parent passes on a single chromosome from each pair. And so each new human has the exact same complement of chromosomes. If all of the chromosomes from a mother and all the chromosomes from a father's cells were combined, then the offspring would have twice the number of chromosomes. So they go from 46 to 92. But then the next generation would double again, and it would double again. So part of what's important about the reproductive system is to keep the number of chromosomes consistent across all generations. So as I was describing the cells dividing and the clonal thing I was talking about, I mentioned kind of briefly under my breath almost that there are a few exceptions to cells being clonally identical to that fertilized egg cell. And one of those exceptions, or two of those exceptions, are cells that are called gametes. Those are the reproductive cells. They carry only one of each chromosome, and when 2 gametes come together, their nuclei combine to give us a full complement of chromosomes. The gametes are the egg cells and the sperm cells that we saw at the beginning. The egg cells come from the woman, and sperm cells come from the man. Like I said, with this karyotype, the XY that we see there tells us that we're looking at a male karyotype. When a sperm cell is generated, or when sperm cells are generated, the chromosomes separate out so that 23 go into one cell and 23 go into another cell. It's a little bit more complex than that, and I'll talk about that in a little bit. But so the X chromosome that we see here would go into one sperm cell, and the Y would go into another sperm cell. And egg cells are always going to have X chromosomes because that's what women have. And so the gender of the offspring is determined by which of these 2 chromosomes the sperm is carrying. So if the sperm carries an X chromosome, when it combines with the egg cell, which necessarily also carries an X chromosome, the resulting offspring is going to be female. And then when the sperm carrying the Y chromosome combines with an egg cell, which is necessarily carrying an X chromosome, the offspring is going to be male. Now, sometimes people get kind of interested in exceptions to the rules and that sort of thing. There are imperfections in the processes that I'm talking about today, but I don't want to get into that, especially when it comes to the sex chromosomes. But it is possible for pairs of chromosomes not to segregate out, and so one gamete might actually carry both sex chromosomes. And if it's an if it's an egg cell, then there'll be an extra X chromosome in the offspring. And if it's, well, I guess I should say, either way, there's going to be an extra X chromosome, whether it's contributed by the mother or the father. But you could have XXY, I guess there's not really a triple X system. But sometimes these sorts of things can sort irregularly and cause problems. At the level of fertilization, when the egg cell and the sperm cell combine, if there are chromosomal problems, oftentimes they will shut down fertilization, and the egg cell will never mature and start dividing because it doesn't have the right chromosomes in it. The kind of chromosome problems we have are things like trisomy 21, which means 3 chromosome 21s, which, like I said, are actually the shortest of the chromosomes. And in that situation, that's what leads to Down syndrome. So there's a little bit more DNA on hand than there should be, but probably not enough to really throw it off. I think an X chromosome carrying sperm cell combining with an egg cell with 2 X chromosomes is never going to go through fertilization. I'm not familiar with that particular situation. I think anything as big as this chromosome here isn't likely to get by the systems that are checking for weird chromosome irregularities. It's short ones like 21, or there are situations where 2 Y chromosomes begin to mix. But other than just mentioning those things, I don't want to really go into that. If you're interested, you can look things up on the internet. Just be careful when you start searching for things like sex chromosomes on Google, because you never know what you might get. But I want to take a little bit of a tangent here to talk about some issues to help us sort of understand species and some reproductive function. So this, like I said, is a karyotype. This is what we would get taking a picture of a cell that's undergoing probably mitosis, which is much more common than the alternate, which is how gametes are made. And in the old days, karyotypes were done in a very manual fashion. So a photograph was taken, a physical picture was taken of a cell undergoing cell division, and then with scissors, each individual chromosome was cut out of the picture, and then they were glued or taped or whatever down to another sheet of paper in this sort of fashion. And you can look at the chromosomes and tell which ones matched up, and you can get all the pairs there. That sort of approach would have been done with black and white photography, and the fact that this is colored tells us right off the bat that this is digital. So this karyotype was probably made digitally, which would be just taking a digital image of something and then excuse me, selecting out the chromosomes and cutting and pasting them into another picture so you can arrange them like this. So that's probably the sort of thing that went on here. But this picture is actually from the cover page, the cover of a magazine, which obviously is called the American Biology Teacher. And these are representations of chromosomes also. These are not taken from a karyotype, but rather they're chromosome maps. So when chromosomes are sequenced, we can draw maps of them based on the sequence of base pairs that we find, and the relative amount of different nucleotides in the DNA leads to this banding pattern as it represents that. What this magazine article is looking at is comparing 4 different species and looking at their chromosomes. The 4 different species here are all great apes. The first one is human, and then the other three, I'm never entirely sure, I don't remember the paper that closely, but it's probably chimpanzee, gorilla, and I don't know, bonobo or something like that. Different great apes. And you can see from the banding pattern that's sort of repeated between the 4 chromosomes in each group, how there's a lot of similarity. But there are some differences, like the human chromosome, which is the first one, we can see these bands all line up, and we can see at the sort of centroid of the chromosome how those would all sort of be at the same point. But there's a little extra sort of inserted into the human chromosome here, which is part of what defines the difference between humans and these other species. Or whatever species this is here, we can see beyond this last band that all the others have, there's a little bit more sort of at the tag end here. There's some other differences that you can kind of see qualitatively. The numbers underneath, 1, 2, 3, 4, is corresponding to the human chromosomes. So this is human chromosome number 1, and then the analogous chromosomes in the other 3 species. And we can see how there's a lot of similarity between them. Or this is chromosome number 2 in the human, and then in these other species, none of them have a single chromosome that corresponds to our chromosome 2. Instead, we can see that there's 1 chromosome in each species with banding patterns suggesting that it's very similar to this end of chromosome 2. And then each species has another chromosome with banding patterns suggesting that they're very similar to this end of chromosome number 2. And that suggests likely what happened, say this next pair here is from chimpanzees. These 2 chromosomes merged together to make one big chromosome. And that's what we see here as chromosome number 2 in humans. That would suggest possibly one of the major things that differentiates humans from these other great apes is just this big chromosomal change. And then if you look across the other chromosomes through this picture, other than chromosome 2, not really corresponding to a single chromosome, the other organisms, but rather these pair that must have merged together. The rest of them are pretty similar across the different species. You can see some changes. So like here, the human and chimp chromosomes are kind of similar, but differ kind of the same way from whatever these other 2 species are here, that gorilla and bonobo or orangutan or whatever they are. So you can kind of look over all of this to see that there's some real similarities between these species, but there's some significant differences. And the biggest difference really is that all of these other species have actually 24 pairs of chromosomes, and humans have 23. Now, part of why I got kind of interested in this stuff is there's a show which was on, I don't know, Discovery Network or something like that, more than 10 years ago. I can't remember how long exactly ago it was, but I don't know if it was Discovery or History or whatever, one of those cable networks which show kind of iffy educational kind of programs. There's a 2-hour show that was called Humanzy, and there was this animal that they spent an hour and a half of that 2-hour show trying to make you think it was a human chimpanzee hybrid. And the organism had some characteristics of it that looked chimpanzee-like, but also had some characteristics, especially behavioral ones, that seemed rather human. It walked upright, it had a bald head. It seemed to mimic a lot of behaviors that we would associate with humans. Also, it did not really interact with other chimpanzees. And they explained this might have been because they just had a different smell to it. organisms associating with each other are used to a particular smell that you would associate with that species. If you have pets, you're probably familiar with that. If you have dogs or cats or something like that, there's a dog smell or a cat smell you're kind of used to. And within a species, all the individuals in the species are probably used to that smell. And so when another species comes along and it smells differently, they know right off that it's a different type of organism. And that's what they're suggesting. And they went on for a long time explaining why there might be some, this might be a hybrid, and there are some really kind of questionable parts of the show suggesting how it could have happened. It wouldn't have been a laboratory, but rather a human and a chimpanzee would have had to mate. So it got rather sensationalistic for a while. But then in the last half hour, what they got to were starting to point out that there are just some things about this organism that were obviously not human. And then they got down to the chromosomes. And if it were a human chimpanzee chromosome, hybrid, it would have to have an odd number of chromosomes, because the human gamete would contribute 23 chromosomes and the chimpanzee gamete would contribute 24 chromosomes, giving us 47 instead of 46, which we'd expect in a human, or 48, which we'd expect in a chimpanzee. And in this organism, when they looked at the DNA, it had 48 chromosomes. So it was definitely not human. It was definitely a chimpanzee or some sort of great ape. It's possible that it was a variant of a chimpanzee that was isolated in a certain area and had sort of diverged from chimpanzees, but not established a separate species, because whatever group it came from might not have survived enough to populate as a whole separate species. It might have just been a little offshoot. But it does kind of get at thinking about some ideas that have to do with hybridization. And so I mentioned a little bit ago some of the problems that we sometimes see with chromosomes duplicating in humans, like with Down syndromes or some of the sex chromosomes issues. And some of them just aren't going to come to term because there's processes happening in the cells when they come together that it just wouldn't allow for that. And really, a human chimpanzee hybrid is probably impossible because of that odd number of chromosomes. The fact that there weren't suggest to the egg that there's something wrong and it wouldn't continue on and divide and become viable. There are, however, hybrids that do become viable. And I have this picture here to show you what I think is just one hybrid. I'm not quite sure what this cat on the back is, but this is what's known as a liger. It's the offspring of a male lion and a female tiger. I grabbed another picture of this because this is a much more impressive picture to highlight. Ligers are huge cats. They're probably the biggest cat around. And part of the reason for that is that the gene that controls growth and body size isn't present in a liger. So it just keeps growing out of control, and they get to be really big. So I like this picture because there's a liger standing up quite high next to its handlers who, I can't necessarily say, if they're average size males or anything like that. But the picture here is really kind of hard to figure out, because this woman here, who, if you look up liger and look up pictures, you'll find several of her because she's with some outfit that has a liger. She usually has dark hair, but she's a fairly petite woman, which really kind of makes this picture less obviously, or no, sort of accentuates the difference here. But still, ligers are huge cats. Now, I wasn't able to find a good source that really tells me what this cat here is, whether it's another hybrid or if it's a different type of, completely different type of hybrid. Now, the fact that this thing's called a liger and it's specifically a male lion and a female tiger mating gets to the fact that there's a different type of hybrid, which is called a tigon, which is a male tiger and a female lion that are mating. I was trying to pull up some pictures, which is where I got the one I just showed you. And I'm not going to pull them up, but in searching for liger and tigon, there are also pictures of mating between a male lion and a female tiger, or a male tiger and a female lion, which kind of gets at the point there. The interesting thing, getting back to the growth, is the fact that the liger grows uncontrollably large because it doesn't have a gene to regulate growth gets at the fact that the gene to regulate growth in lions is found in the female genome, and to regulate growth in tigers is found in the male genome. So when a male lion and a female tiger mate, neither of them contributes the appropriate gene to regulate growth. The alternate tigon is actually a fairly small cat. I don't think this is a tigon because it wouldn't be that small. This is a cub of some sort. If anything, it kind of looks like a liger cub. But a tigon is going to be a small cat relative to a lion, a tiger, or a liger, but not like a house cat kind of small cat. Just its growth will be limited quite a bit. Now, if you look up hybrids, you can maybe find some other suggestions of types of things. I was doing that earlier and I didn't pull up any pictures because too often what comes up in a Google search for hybrid are Photoshop creatures. So I didn't want to go there, but this is a real organism. And what I was showing you a second ago was too, the one that was standing up. We call these hybrids because we consider lions and tigers separate species. And part of what defines them as separate species is just geography. They probably have the same chromosomes. So genetically speaking, calling them separate species is a little bit hard to nail down. That's not the only definition. Also, lions and tigers are found in different geographical locations, so they're not going to interbreed. As I was going through this stuff, I actually found a note that suggests that possibly at one time ligers and tigons were naturally occurring species because before human civilization expanded, there was an overlap in the ranges that these organisms are found in. But they aren't viable species. They don't have offspring. Is that what you're going to ask?
Kind of, yeah, I have a question. So I know that like when horses and donkeys mate, like I don't remember which one it is. It's either a mule or the other one. One of them comes out and they can't reproduce.
Right.
So I think that's what
yeah. So if you want a mule, you have to get a horse and a donkey to mate. And again, there's gender issues involved. Male horse, female donkey, probably. And then the opposite has a different name, which I want to call a hinny. I don't know if that's the right word for it, but anyways. As far as I know, both of them are actually sterile. So if you need a new mule, you don't get two mules and have a mule baby. That doesn't work. Instead, you have to take another horse, another donkey, and get things started with them. And with the cats, the ligers and the tigons, they don't seem to be viable organisms either. They can't reproduce. They tend to be infertile, which gets at another idea of that there might be actually some species separation between lions and tigers. But what really defines species gets a little complicated, and I don't want to get into that because we're really just thinking about the one species of humans. And humans are the only species that have 46 chromosomes, or I should say the only species that I've found that has 46 chromosomes. There might be other species that do, but are very unlikely to be able to hybridize with humans. It might be there's a plant or something like that that has 46 chromosomes too, and I just haven't run across it yet. But with the other organisms that humans could theoretically hybridize with, there's not a match in chromosomes, so it's not likely to happen. Now, I was talking about mitosis earlier, and I was talking about the production of gametes. So I want to talk about cell division a little bit. And the two types of cell division are mitosis, which I talked about before, and the other one, which is meiosis, which is what gives rise to gametes. They both have very similar processes. It's just that there's sort of twice as much stuff going on in meiosis. So in these pictures, the chromosomes are represented in these colored structures that are paired up. So we have a black and a gray and a red and an orange and a dark blue and a light blue, I guess. When we say chromosome, this is what we really mean. One of these is an independent piece of DNA. But before the cell goes through my division, the chromosomes duplicate. The DNA makes a copy of itself. So now we have these more traditional looking X shapes here. And actually, if we go back up to these, you can kind of see that there's two arms of each chromosome sticking off here and two there. So it has that kind of X shape that's suggested here. So the chromosomes duplicate. And then when the cell is going to divide, half of each duplicated chromosome separates out to give rise to the two offspring cells. And then what we see down here is an identical copy of the original, okay, or two identical copies of the original. What happens in meiosis is a little bit different. There's also duplication that takes place. So the single chromosomes that we see up here end up having two copies, but then the paired chromosomes also come together, forming what's called a tetrad. And what happens in tetrad formation, which isn't illustrated in this picture, but the word synapsis gets at what's happening, is little chunks of DNA from one chromosome will swap with chunks of DNA from its paired chromosome. So they could represent that in this picture if they took a little bit of the black end of this and a little bit of the gray end of this and swapped it. And that's exactly what happens. A little bit of DNA from one side crosses over with the other, and the DNA just sort of swaps chromosomes. It mixes up the DNA a little bit, and it can provide a little bit extra variation in the genes that get passed down. But that's what synapsis is, although it's not really represented here. The tetrads then separate out, giving us two cells that each have the duplicate of chromosomes there. But we see the black chromosome on one side, the gray chromosome on the other. Red on one side, orange on the other. Dark blue on one side, light blue on the other. So we've actually separated the chromosomes out. So instead of the three pairs, what we have here are three individual chromosomes that happen to each be duplicated. And that's what we get to at the end of one division, the first part of meiosis. And then those two cells that each have one of each of the chromosomes, but in a duplicated form, they separate or divide into two cells without any further DNA duplication. So just the duplicated DNA that we see here splits, so half of it goes on to one cell and half of it goes to the other cell. And we get four cells that each have three individual chromosomes compared to the three pairs of the original up here. Okay. And then if we take two gametes, not two from the same set, but if we take a gamete from the mother and a gamete from the father, they're each going to contribute one chromosome from each pair, and then the resulting fertilized egg will have a full complement of chromosomes again. Okay. Now in these pictures, to keep them somewhat simple, they're just showing us three chromosome pairs, but really what would happen For humans, is we'd be looking at 23 chromosome pairs, which would be really hard to illustrate in these pictures. The point, however, is to get to making these gametes, which are what are going to be passed on from generation to generation. And when they combine, they make a new individual with the correct number of chromosomes, except for those few cases where things go wrong. But it is possible for chromosomes, as they're segregating out in one of these steps, for two to go one way and none to go the other way, and you get extra chromosomes in some of the gametes. It can happen. So now, I think in the presentation it goes on to start showing some of the structure. Yeah, that's from another book. So what I want to do is move to the book and talk a little bit about the structure of the male reproductive system. And in lecture, we're going to look at the structure and function of the female reproductive system. What I want to do with the male reproductive system is basically follow along the path of the male reproductive tract from where the sperm are made to where they end up being released. Oops. The first section of the book here is anatomy and physiology of the male system. Some of the physiology of the system I'm going to talk about in lecture, just as it fits in with the female reproductive anatomy, physiology. So this picture kind of shows us all of the components of the male reproductive tract. And I'll come back to this picture to point things out. We start with the testis, which is the male reproductive gland. This is where spermatogenesis takes place. This is the formation of the gametes. Okay. And this is where we want to start looking at the reproductive tract for humans. The testis is actually made up of tubes, or made up of structures called seminiferous tubules. And the wall of the tubule is where that spermatogenesis takes place. So here's a picture in the textbook of the testis, and it's really just a whole bunch of tubes. They're organized into little regions separated by connective tissue septa, but it's really just a bunch of tubes where the meiotic divisions take place. So the cells that we can see making up the wall of this tubule here are mostly cells involved in this process. The stem cell that gets to start off, called a spermatogonium, has 46 chromosomes, which is represented by saying 2n there, which means that there's twice the number of chromosomes. So there's pairs of everything. It divides, and one of the cells that comes out of that division is the primary spermatocyte. The other one cycles back as the stem cell, which is a spermatogonium. So it's continually sort of generating a primary spermatocyte, which is a copy of the spermatogonium, again, through mitosis. Then this cell goes through meiosis. It duplicates the DNA and segregates into two different cells called the secondary spermatocytes. And these are designated by 1N, or just as easily could be N, which means that there's not pairs of chromosomes here. Rather, there's just one of each chromosome. The N there represents the number of chromosomes, which for humans would be 23. So 2N would be 2 times 23, or 46, and 1N would be 1 times 23, or 23. So the stem cell and the primary spermatocyte have 46 chromosomes in both of them. And then half of those chromosomes go into either of the secondary spermatocytes. So we've now separated these two cells that have half the number of chromosomes. The chromosomes are still duplicated at this point. So the duplicated chromosomes then split, giving us two gametes, which are called spermatids, from each secondary spermatocyte. So now at the end, these cells are the ones that look like half of the original cell. After spermatids are made, after the gametes are completely divided out, then they start to change into the spermatozoa, or the sperm cells, which is they shed a lot of their cytoplasm, so they're really just DNA delivery cells. So there's just the nucleus in the center here, and then this kind of collar at the end has a bunch of mitochondria in it to power the flagella to move for swimming. Okay. So we see the specific cells there. I want to point out in this picture, there are cells called Leydig cells, or interstitial cells, in the tissue outside of the tubule. And then there are Sertoli cells, which the book refers to as sustentacular cells. You'll also see those called nurse cells. They make up the wall of this tubule, and they nurse these cells through the process of division. So what I'm going past fairly quickly is where it's mentioning the Sertoli cells and that sort of thing. But back to, this is the testis itself. The tubules coming out of the testis then come together and then all feed into this structure here that's still a bunch of tubules called the epididymis, okay, which we see here. We're not looking inside of it, so we don't see all the tubules, but this sort of worm-like thing laying on top of the testis is the epididymis. So the reproductive tract, the tubules, lead into the epididymis, and then the epididymis sort of condenses down into a single tubule, which gives rise to the ductus deferens, which you've probably heard of before as the vas deferens, but ductus is its more appropriate term. So this is the tubule that carries sperm out of the scrotum into the pelvis. Go back to this picture here. So the testis again with the epididymis on top of it, and then coming out of the base of the epididymis is the ductus deferens, or vas deferens. Now, when we call something a vas, we're referring to it as being a vessel. And vessels, by definition, have a simple squamous epithelial wall. The reason why this isn't actually the vas deferens, but rather the ductus deferens, is because it does not have a simple squamous epithelium. It has a different epithelium, which we would find in a duct. And that's where the name difference actually comes from. The ductus deferens extends out of the scrotum and into the pelvic space as it's sort of looping over the pelvic bone here. It travels alongside the the bladder, and it meets up with this structure here, which is called the seminal vesicle. So on the posterior bladder, that's where we see the ductus deferens ending in its ampulla, which meets up with the seminal vesicle. Now, the ductus deferens has been carrying the sperm along. The seminal vesicle is where we start to see some of the fluid that's going to contain the sperm produced and added to the reproductive tract. Okay. So this is the first structure that makes seminal fluid. Okay. It's called the seminal vesicle because it's where seminal fluid is made, which is again going to be the fluid that the sperm is contained within. The next part of the tract is the urethra. So the ductus, oh, sorry, not the urethra, getting ahead of myself. The ejaculatory duct. The ductus deferens and seminal vesicle meet together within the prostate gland as a common duct, okay, the ejaculatory duct. So going back to the picture here. So where the two come together here, the duct that continues on from there is the ejaculatory duct. There's one on either side, but they're very, very short, and they're just passing within the prostate. Then they meet up with the urethra. As they're passing through the prostate gland is making more of the seminal fluid, or what we would call the ejaculate. Again, the fluid that the sperm are suspended within. Then we meet up with the urethra. Where the ejaculatory duct merges with the urinary tract. And we see the in the book they make a point of the urethra from the bladder down just colored white because that's the urinary tract. But then when we get to this point where the ejaculatory duct meets up with the urethra, from that point on, the urinary and reproductive tracts coincide. And they're pointing to this area and specifically calling it the prostatic urethra, meaning that it's the urethra going through the prostate. It doesn't necessarily mean it's just that part right there. It's all of the urethra as it goes down through the prostate is the prostatic urethra. But that's where the ejaculatory duct meets up with it. The urethra then extends through the rest of makes up the rest of the reproductive tract and extends through the length of the penis where it opens to the outside world at the external urethral opening, or some sources will call it the external urethral orifice. And that's just the end of the pathway. Now, there's another gland here that contributes to the pathway. called the bulbourethral gland. At the base of the penis, the bulbourethral gland contributes more fluid to the reproductive tract. This is mostly pre-ejaculatory, helping to neutralize urine pH and clear the reproductive tract. But there's more of that. So let me make this a little smaller. Whoops, smaller, so everything fits on the screen there. But that kind of runs through all of the components of the reproductive tract in the male system. Okay. Oops. So all of that's suggested in this picture down here. At the top, they're just showing some variation in the presence or absence of the foreskin around the head of the penis, and the structure of the penis, the shaft, and then giving into the head or the glands of the penis. See there. Okay. Now, as the urethra goes through the length of the penis, it's surrounded by this rather spongy tissue. There's actually two components to it. In the sagittal view, it looks like there's this rather significant tissue that's directly surrounding the urethra, and then some minor tissue at the top here that's running along the length of the penis. But actually, what's surrounding the urethra is kind of the smaller part, which is called the corpus spongiosum. And then on midline, we don't see all of it, but sort of superior to or dorsal to the corpus spongiosum, the other tissue is called the corpora cavernosa. Now, there's two corpora cavernosa. So in Latin, this is a plurality. Corpus cavernosum would be the name of a singular one, but we usually refer to them as a pair. They have running down the length of them major arteries, which will bring blood into this tissue. And as the tissue engorges with blood, that's what causes the actual erection that we see there. Looking at this tissue under the microscope, you can kind of orient yourself to this, because actually under the microscope, it really looks a lot like a face. The urethra is going to be collapsed, so it'll look sort of, more like lips than this picture suggests. And then the center of the corpora cavernosa are going to have these arteries, which will look kind of like the pupils in the middle of eyeballs. So that'll help you kind of orient to what the different structures are there as you're looking at it in the microscope. Speaking of looking at things under the microscope, that's what I want you to look at today. So I want you to go through the reproductive tract and look at these structures under the microscope. And especially I want you to pay attention to the epithelial lining of the tract. I've actually specifically not said what epithelium is lining the tract, because what I want you to do is, looking at these under the microscope, figure out what epithelium is common throughout the male reproductive tract. And I'll give you a little direction on where to look for that. But you will have a question on one or more of your assignments, which might be the lab quiz, might be some of the part 1 or 2 questions, or you might see it on the final, depending on if you get that question, about what tissue makes up the male reproductive tract. So I want you to look at these slides in the blue box, starting with slide number 5, which is the testis. And so you'll see the seminiferous tubules where spermatogenesis is taking place. And then 6 is the epididymis. 7 is labeled on the slide as the vas deferens, but of course it's the ductus deferens. And then 8 is the seminal vesicle. And 9 is the prostate gland. And then 10 is a cross-section of the penis, which is sort of like I was talking about a second ago. You can see the corpus spongiosum and the corpora cavernosa, and with the urethra, it kind of has a face-like appearance. And you'll see that in the cross-section of the penis. Now, it'll be really hard to figure out the epithelium in the testis because of spermatogenesis. There's a lot of extra cells in there, which really kind of confuses what's going on there. But in 6, 7, and 8, you definitely should be able to get an idea of what that epithelium is. In the penis, depending on exactly where you're looking, and the sections are going to be from varying positions along the length of the penis, you aren't necessarily going to be looking at that epithelium there. At some point, the urethra changes to different epithelia, so it's not a good source of where to see that. In slide 9 with the prostate gland, you may or may not get part of the prostatic urethra or the ejaculatory ducts. It depends on exactly where the section is taken. So you might be looking more at the glandular structure there. You can see that. So sort of in the spirit of this being the end of the semester, at least as far as lab's concerned, I'm leaving it to you, because I think you should be able to, at this point, figure out what epithelium you find lining the tract as you're looking at it in the microscope. So I'll leave that for you to figure out, and I won't answer a question that leads to answering that. But I will answer any other questions you have as you're looking at this stuff. When you feel comfortable with this material, you're welcome to clean up and head out for the day. And there's lens paper right here if you want to grab it while you're getting stuff. If you happen to notice any of the slide, the scopes when you get them out are put back funny, let me know, because I think that's been happening a little bit in some of the other classes.
What has been happening?
It's the other classes are putting them away wrong.